Hi guys, my name is Manpreet, and this is my friend Sydney, and we're from Martin Lab on this floor. Welcome to Microbial Painting, where science meets art. Have you guys worked with microbes before? Okay, so today we'll be working with yeast and bacteria. They're both non-pathogenic microbes. The yeast we're going to be working with today is called Saccharomyces cerevisiae. It's the regular brewer's baker's yeast that you guys probably have heard of, probably used during baking. And we're just going to use a classic E. coli in the lab. We have 4 colored yeast, white, yellow, orange, and red in the lab, and 3 colored E. coli, white, green, and red. So yeast usually doesn't naturally produce these colors. So what we've done is this is a genetically modified yeast strains. We've taken the carotenoid genes from other plants like lycopenes in tomatoes gives it red pigment, and beta carotenes in carrots gives it orange pigment. We've taken those genes from the plants and introduced them into the yeast such that now when the yeast grows, it expresses those colored pigment proteins. And you can see them under the blue light. For E. coli, we introduced fluorescent proteins so that when E. coli grows, it glows and produces the desired fluorescent protein that was introduced into it. So we add the fluorescent protein on a plasmid, which is a circular DNA, and we replace an undesired gene inside E. coli with our desired fluorescent protein gene in the plasmid. And when the E. coli grows, we look at the colonies to see if it's glowing the desired color. If I introduced green fluorescent protein into the E. coli, when the E. coli grows, I look at the colonies and see if it's green, and same with the red. We've already prepared these for you guys in the lab today. You will be given Petri dishes labeled either black lines or green lines. Black is for yeast, green is for E. coli. You can draw whatever you want on the Petri dish. Sydney will give a little demo on how to do it. Just make sure that you do not mix yeast and bacterial plates because the plates have different nutrients needed for either yeast or bacteria to grow. So if you streak a yeast colony on a bacterial plate, it wouldn't grow, so you won't be able to see your drawing. Some of the safety measures when working in the lab, since this is a working lab, people are still conducting their experiments in the lab as we speak. So make sure you don't touch anything on the shelves or anything that was not on your working station that was told to you to touch. You will be working with an open flame to provide a sterile environment while you're working, because how the flame works is it keeps the air rising upwards. So any airborne contaminants will not fall onto your plate and they won't grow because you don't want that happening. Anyone with long hair should have their hairs tied back, which I think you guys already do. We will give you gloves to wear in the lab. You already have your lab coats on, which is great. And I think Sydney will explain a little bit more about the drawings we're going to do.
Yeah, so I'm just going to grab this here. Sorry, I forgot to grab it to explain. So basically what we're going to do is, like Manpreet mentioned, you have either your bacteria or yeast plate. So you're going to pick one. We only have enough time, unfortunately, to do one or the other. But this is kind of a preview of the colors you can see. So here for the yeast, you have white color. You have kind of this light orange color, orange, and like a reddish color. And then for the bacteria, you have white, green, and red. And so for the bacteria, because as Manpreet mentioned, they're fluorescent. Once you put it under blue light, you can see this is how the fluorescent proteins look. And we are going to incubate them and take pictures and email them back to you so you can see what your drawings look like. So I'll explain in the lab how to actually streak out the plates and how we're going to do that. But for about 5 minutes or so now, there's some stencils there and colors and stuff. So if you want to do a little drawing of what you're going to do, then when we get in the lab, you can put that under the petri dish and draw from there. Just keep in mind that like when we're drawing, we're going to be using either cotton swabs or toothpicks, so it's not going to be super detailed, but that's it. The cotton swabs obviously are a bit thicker. The toothpicks can do a bit of detail, but with the way microbes grow, you can't do like super fine lines. So just keep that in mind. But yeah.
Yeah, and if you guys have any questions, we're going to be here. Feel free to ask anything you want.
Mhm. There's also a little
So we did this activity last year with the CGEP students, and here's some examples of some of the plates that they made. We will take photos of the plates and send it to you in your email.
what they'll look like if you yeah,
Yeah.
so you can mix the different colors as long as they're and it's labeled in there, so you can there's 3 E. coli colors, so you can mix those, and then there's 3 or 4 yeast colors, so you can mix those. Just don't mix the E. coli with the yeast because it won't grow. So some examples of what some of the previous people did, if that gives you an idea of what you can do. So yeah.
Do you guys have any questions?
No.
Oh, all good. If you don't have anything in mind, you can just wing it and
Yeah.
draw whatever you want.
Have any of you ever worked with microbes before? No? Okay, cool. Okay.
Little bit of squeaking.
Okay. Yeah, okay, nice.
With the loops and the sticks.
Yes, okay, perfect. So you have a little preview kind of
I don't know how to streak without grazing the agar yet.
Oh yeah, that's the tricky part. So I'll show you guys in the lab. You don't want to press too hard into it, otherwise it can cut the agar. But with the cotton swabs, it's actually a bit easier because it's kind of you would have to press really hard. But with the toothpicks, it's easy to cut the agar up.
So are we ready? Okay. Yeah, so we're gonna go first and then. Okay.
Okay, so if you guys want to all kind of come around.
We have small and medium gloves here. Everyone please glove up. Also, before we start any work on the working stations, we always sterilize it with 70% ethanol, which we've already done, and we'll do it after the work is done. It's good lab practice to remember.
Mhm.
And now Sydney will
Yeah,
give a
So
demonstration.
I'll show you guys over here. So is this everyone? Oh yeah, okay, no worries. So okay, so what you do to start with is you gotta light the Bunsen burner. So you take this, it's a striker, so it makes a little spark, and you turn on the gas here and light the flame. So when the flame is lit, essentially you have about a 1 foot radius around the flame, and that's your sterile environment. And so that prevents any airborne contaminants from getting into your plates. So here we have, like we mentioned, the 3 black lines are going to be your yeast plates, and the 3 green lines are going to be your bacterial plates. So you can pick one or the other. And what you're going to do is you're going to take a marker and write your name, or just your initials if you want, on the top, so we know whose is whose. And basically you can see here that there's agar in the plate, right? And right now it's actually flipped upside down, but when you flip it over, you take the lid off, and this is where you're going to be streaking, is on the inside there. So what you're going to do, we'll get you a new thing of cotton swabs, but these are the toothpicks. Okay. These are the cotton swabs. So you can pick either or. Like I said, the toothpicks will be a bit more fine. The cotton swabs will be a bit more thick. So you're going to take one out. This. And remember, you don't want to touch anything that's going to be touching the plate. So just don't touch the end that's going to touch the plate. And here's your cultures here. So they're labeled with the color and whether they're E. coli, so for the bacterial plates, or yeast for the yeast plates. So if you take a culture, you're just going to pop it open close to the flame. You can dip your cotton swab or toothpick in the culture. And then you take the lid off and you just touch it to the surface here. So you just kind of go across like this. And of course, in whatever pattern you want, you can do like a little smiley face like this. Just be careful not to press too hard that you're gonna break the plate. So that's pretty much it for that. And then you put the lid back on when you're done. And just a side note, when we incubate them, you won't see this part, but you always flip it upside down so that the agar is facing down, because if you were to incubate them like this, condensation could form on the top and that could drip down onto your plate and contaminate it. So always like this once everything's done. And you can actually see the little streaks on it, so you can have an idea of what you did. So yeah, any questions before we start? No? Okay, so we can do 2 at one station and 1 at the other. So you can just go on either side of the flame here.
And once you guys are done, please put your gloves in the biohazard waste over there, the yellow bin.
Don't throw your gloves out in the normal garbage. That's it. Okay.
And yeah, if you have any questions, just let us know.
Thank you.
Yeah, it's nice. Good job. No, that's perfect.
Also, if you accidentally double dip or you think you contaminated a culture, please let us know so we can give you a new one.
Mm-hmm. Yeah, I forgot to say that. Oops.
Oh, I'll try and answer.
That's green. So GFP stands for green fluorescent protein. Yeah, and then the red one is sometimes called RFP.
Oh, I'm sorry, I wrote GFP on
Oh,
it.
that's okay.
I do put on the blacks for you.
Yeah, so once you dip it once, you can throw it out just in the bucket there.
Yeah.
And then take a new one to dip it again if you want more of the same color or a different color. And we have more too if you run out, so just let us know. That's okay. That's kind of how science seems to go. Yeah. Yep, exactly. Yeah. Awesome. And then just write your initials on the top here. Yeah. Or if you already wrote on the
Done.
bottom, it's okay. Actually, maybe just do it again. Yeah, no. Awesome. Cool. Yeah.
Sorry, where did you say
The gloves go. Yeah, so just there's a yellow bin there with the yellow garbage bag, just in there. Awesome. How's it going?
Good.
Nice.
That's more like an experimental, like.
Nice. That's cool. That's like a cool.
How long is the chip that stuff's that?
Yeah, so you just you can see on the toothpicks, it's just that small like, yellow one there. So here I can show you like an example if you want. So you can kind of you can see that there's like the end that's sort of not so pointy. So dip it in. And you don't want to go like this. You kind of just want to use the side and just touch like very lightly at first to kind of get a feel for it. And then just you have to go like very light because if you go like even a little bit, you stab it,
Yeah.
see, like that. So yeah, just very light kind of on the surface like that, and you'll see your streak. Yeah, awesome.
Stressful.
I know, yeah.
No, it's not the camera, there's the flame, honestly.
Yeah, no, it's sure. Yeah, I guess after a while you get a little bit more used to working with the flame. There's also you can work in a sterile hood too.
Yeah.
So
Okay, well, that might make it.
Okay, I'll take the plates. Thank you so much, guys.
Yeah.
That's everything.
Um,
Hope you guys had fun.
yeah.
Bye.
Thank you.
Yeah, thank you. Okay, cool.
Don't forget to wash your hands.
Okay, thank you to everyone who joined us for this lab walk. Thank you to those in person, those at the workshop, and those online. We're going to be closing up the live stream now, but a quick reminder that this conversation, this lab walk, is already available on our YouTube channel if you'd like to revisit or share it. Please join us once again at about 1:15 p.m. for a keynote. All right, everyone, have a great afternoon.